Hello and welcome. My name is Andrea Tanzer, and today I will present to you my latest work on the evolutionary conservation of RNA editing in filamin genes. So RNA editing is a process that defines the epitranscriptome and forms yet another layer of post-transcriptional regulation. In this process, the identity of nucleobases is chemically altered by enzymes. And these resulting RNA modifications can be considered as temporal SNPs on the transcriptome level. These modifications affect all downstream processes such as splicing, translation, in general RNA protein and RNA-RNA interactions, and can also lead to recoding events. That's the change of a codon for another amino acid. In my study, I'm focusing here on a process called A to I editing. And here, adenine is deaminated, leading to hypoxanthine. So adenosine then becomes the nucleotide inosine. This process is catalyzed by ADAs, adenosine deaminases acting on RNA, which are proteins consisting of double-stranded RNA binding domains, a deaminase domain, and one family member, ADA1, also contains said DNA binding domains. The double-stranded RNA binding domains of ADAs interact with the double-stranded RNA substrate, where the adenosine in question is flipped out into the deaminase domain and where the catalytic reaction takes place. In general, A to I editing and ADAs have been found throughout animals and animals only. So in this project, we are trying to understand the functional relationship between RNA modification and the respective RNA structures. And to do so, we study the genome in general and selected genes. For instance, the filamin genes, which harbor an RNA editing site in an exon 42, which leads to a recoding event from Q to R. And the respective target structure has been proposed to be formed between the exon and the start of the intron, as well as a further downstream region in the intron which resides in an ultra-conserved element, giving rise to a double-stranded RNA structure which is recognized by ADA. So we wanted to know whether these editing sites and the respective target structures are conserved, or whether in different animals, if the editing exists, is performed through different ADA target structures. In order to do that, we set out to identify filamin genes throughout all the animal vertebrate genomes available, and we selected the selected NCBI reference genomes, at the time 1,363 species. And found that the filamin A and B, or confirmed that filamin A and B, those copies that are edited, are more closely related than filamin A and B to C. What we also found is that the exon structure is conserved, and not only the exon edited plus the two upstream and downstream exons, also the ultra-conserved element is preserved in filamin A and B throughout vertebrates. We then used the sequences identified here to calculate multiple sequence alignments of the filamin genes. And from those multiple sequence alignments, we calculated consensus RNA structures to see whether or not the target structures of ADAR, the potential target structures of ADAR, are preserved. And indeed, what we found is that not only within filamin A and filamin B the target structures are conserved, but they are highly similar to one another. And when combining those sequences in multiple sequence alignments, we see that the core motif that is recognized by ADAR indeed is conserved. So with our study, we show that the RNA editing site in filamin A and B is conserved all the way back to early vertebrates, from human to shark and even into lamprey. When we got those results, we were very excited, and of course we wanted to know whether these editing sites could be functional. So we looked through RNA-seq data sets and indeed found that in Danio, the editing site in filamin A and B is used in different tissues at different rates, suggesting that the events we are seeing here are not genomic SNPs, but indeed RNA editing events. And looking further, we confirmed the functionality of our predicted editing sites in frog, in shark, and also in lamprey. To summarize, what I showed you here today is that the filamins A, B, and C arose during vertebrate radiation, the 2 rounds of consecutive genome duplications in vertebrate evolution. For the ADAR target sites in filamin A and B, we showed that the sites are conserved in structure as well as in sequence. And we found strong experimental evidence that filamin is edited from human to mouse and fish into early vertebrates. Thus, the editing site we have studied here is approximately 750 million years old. However, the function of this recoding event remains largely unknown. This brings me to the acknowledgements. The work I was presenting here today has been mostly conducted by Gerhard Gruber and Florian Stalowitz, 2 bachelor students of mine who finished their thesis meanwhile. Anja has been working on other aspects of RNA modification in general, and Sonja has been looking into sequence motifs, and these works will be presented at a different venue. The funding for this project comes from the Austrian Science Fund, FWF. I thank the organizers for selecting my work here for presentation, and I thank you very much for your attention.